Next speakers will be Mr. Snehashish, and he will present the title of Zoonotic Potential of Group B Streptococcus in Aquaculture. Mr. Snehashish is currently a PhD candidate at University Putra, Malaysia. He received his master's degree in zoology from Banaras Hindu University, India. He is currently working on pathology and pathogenesis of Group B Streptococcus serotype 3 ST283 in tilapia and human cells line models in University Putra, Malaysia. Now the floor is yours.
Greetings to one and all present over here. My name is Snehashish, and the topic of my poster presentation is Zoonotic Potential of Group B Streptococcus in Aquaculture, a short review. This poster is authored by myself, Snehashish, along with Dr. Ina Salwany, Mohammad Yasin, Dr. Syafinaz, Amin, Nordin, and Dr. Mohammad, Noor, Amal, Azmai. Dr. Mohammad, Noor, Amal, Azmai is also our corresponding author. Our affiliations and our associations are mentioned here. Kindly refer to that. So, beginning with the topic, when we talk about diseases in aquaculture, zoonosis doesn't come up very often. However, it should. One of the most aggressive infectious agents in the aquaculture industry are bacteria, and hence focusing on them and creating a space of knowledge about them is our primary goal. So beginning with an introduction, a brief introduction, Group B Streptococcus or GBS is a major pathogen in fishes and is a menace to the aquaculture industry. It is a leading cause of neonatal infectious diseases as well as a growing cause of diseases in non-pregnant adult humans. Some subpopulation of GBSs also have been reported to get transmitted from fish to humans and hence hold a zoonotic potential. Understanding on the transmission of GBS in food fishes and humans may prove vital in connecting the links that will explain how the zoonotic spillover occurs. So considering the fact that the subject here is Group B Streptococcus, Group B Streptococcus or Streptococcus agalactiae is the only representative of the Landsfield classification Group B in the group of Streptococci. This is the only Streptococcus species that is found in the Group B, and hence we use the term alternatively as GBS or Streptococcus agalactiae. Coming on to the GBS itself, GBSs or Group B Streptococci are gram-positive bacteria. They are non-motile and form chains or pairs when we suspend them in liquid medium. Group B Streptococci are positive for beta hemolysis. They show a clear beta hemolytic streak when they're streaked on blood agar. They test positive for the CAMP factor, and biochemically speaking, they are catalase and oxidase negative. While GBS population has several subpopulations of its own, we are going to focus on one particular one which holds the most zoonotic potential out of them. GBS serotype 3 ST283 is a unique subpopulation of GBS, as it has an unusually high rate of infection and it is very invasive, both for fishes and humans. This is the primary pathogen that was discovered to be the reason for the outbreak of GBS in 2015 in Singapore. What was peculiar about this infection was that this is the only recorded food-borne GBS infection in the history of mankind. Primarily speaking, the GBS serotype 3 ST283 was classified based on the capsular typing, which basically is the constitutive polysaccharides on the surface of the bacteria, which put it in the serotype 3. And based on the multi-locus sequence typing analysis, this group was further classified into the ST283 group. So hence GBS ST283 was the entire nomenclature of this strain. Back in 2015, this bacteria was this bacteria caused a major outbreak in Singapore, which was foodborne, and the major causative was found out to be the consumption of infected raw fish meat, which was served in the form of a traditional salad, which is a very common practice there in Singapore. However, this is not endemic to Singapore. It's basically pan Southeast Asia, and hence we will take a look at the epidemiology of GBS ST283 or zoonotic GBSs in general. So the cases of zoonotic GBSs especially come from the Southeast Asia region. The primary countries that have shown positive reports for such transmission of GBS from fish to human are China, Malaysia, Singapore, Indonesia, Vietnam, Cambodia, Laos, Indonesia, etc. The list is far longer. However, these are the countries from where confirmed results were obtained. So now coming to the transmission side of this disease. GBSs are, since they are a fish-borne pathogen, they get transmitted amongst fish through fecal-oral route. So that means the infected fishes in an aquacultural setup, they shed this bacteria in their feces, and the other fishes, they ingest the bacteria and get infected. And this cycle just keeps on going. However, the transmission from fish to humans is still a matter of research, because confirmatory results still do not, better spoken, clear-cut results have not been posted about the transmission from fish to human. However, we have reports that suggest that consumption of infected raw fish meat was the primary source of GBS infection in humans who got the zoonotic variant. And also, second, certain reports also suggest that handling of the infected viscera of fish without proper hygienic protection can also be another reason from which humans can come in contact. In the Southeast Asian context, when we see, or even in the Central to South Asian to Southeast Asian context, majority of the populations are fish-consuming populations, and hence the aquaculture industries are very large. The issue comes in when lower standards of hygiene are practiced, and at places like that, such transmissions can be easily overlooked, and they may enter the population very well. However, reports also suggest that consumption of raw meat is not the only form from which this bacteria can be introduced. Hence, higher standards of hygiene while packaging of fishes is absolutely of paramount interest. So when we look at the fish itself that gets infected, so this is a tilapia, and this was infected with the GBS. So the manifestations come in the form of exophthalmia, which is popping out of the eye, and the eye turns a very opaque whitish color. Then we see the skin erosion, which is basically patches of sloughed off skin where the tender infected epidermis is visible. And this also becomes a site for secondary infection, which comes later. And a third major symptom is hemorrhagic patches on the caudal fin. So like when we look at the caudal fin, there are like blood-filled patches on the tail. And one of the later signs of this infection is also distension of the belly. That is, the belly gets filled with fluids because of the disturbances that happen during osmoregulation, etc. When we look at the human side of things, when we look at the manifestation of disease in humans, the primary symptoms are very mild. They can be in the form of just gastrointestinal distress. They could be in the form of fevers, etc. However, since the zoonotic GBS or the GBS ST283 is a hyperinvasive one, it has, when the infection progresses, it has much more dire consequences. The dire consequences manifest themselves in the form of meningitis, endocarditis, sepsis, spinal infection, and joint infection. While in the case of joint infection, there's one treatment that has saved lives is amputation, which is not exactly a treatment, but it's a way to save the life of the patient who is infected. However, when it comes to extreme infections like meningitis and extreme sepsis, patient life is at danger. GBSs come from various hosts. They are very versatile. They're very cosmopolitan, especially in a farming setup, not just aquaculturally. They're there around humans in many different forms. So some of the well-known hosts of GBS have been mentioned in this small diagram here. One is fishes, second is humans, third is frogs, fourth is rabbit, fifth is bovine, and sixth is dogs. Reports of other amphibians and reptiles carrying the GBSs also exist, but these are the sources which come up close to humans very often. While ST283 or the zoonotic GBSs have been reported in the case of fish, humans, and frogs. However, confirmatory identification of zoonotic GBSs have also been done in marine mammals. So finally wrapping this entire point together, the point of this entire poster was to create awareness about a bacterial infection which holds a zoonotic potential and which can have, which has the potential of becoming a major outbreak like it did in Singapore in 2015. However, if the hygiene standards and if the standards of aquacultural disease management, etc., are not evolved well, then we may have a bigger outbreak in future. Secondarily speaking, that since there is a zoonotic potential, then developing control measures, developing vaccinations, developing therapeutics on the fish level as well as the human level hold paramount interest. However, as aquaculturalists around the world and as people from fish diseases and infection biology, I believe that that the way to go forward is to put more and more attention to this disease and design as many control measures and therapeutic options as we can for an infection like this, especially considering the technologically challenged and to a certain extent economically backwards countries like the countries and providing them with alternatives which are economical, however, are effective in controlling the infection in the population. Thank you very much for this great opportunity to the DAA11 Secretariat, to the DAA11 Organizing Committee, to Dr. Amal for supervising me through this entire poster. And thank you so much for this conference. And that's it from me. Thank you.
Thank you, Mr. Snehashish. Can you answer the question? Okay, you're on the Zoom now.
Yes.
So in the chat box, Edliano asked if the infected fish shed the bacteria through feces. How can handling be a potential route?
Thank you so much, Ms. Speaker. I'd like to answer the question in two different parts. So the first part is if infected fish shed the bacteria through feces, which indicates systemic infection, how can handling be a potential route of infection? So one is the shedding of the infection as far as the analysis goes, as far as the studies that have been carried out by several labs, including Dr. Amal's lab, even in labs that are in South America, etc. They have repeatedly reported that one of the major routes through which GBS gets shedded from a fish is through the fecal oral route. The fish which have developed an advanced systemic infection, the defecate and the fecal matter contains shows positive for the bacteria, and that is how when the other fish consume it, they acquire the infection. While there's also another major source of infection amongst fish is that when in an aquacultural setup, infected fish are, for example, if they're ignored, those corpses may become a source of food for other fish, and that is how also the fish can acquire infection. Second is you're asking how handling can be a potential route for infection in fish, infection from fish to human. So that point comes in order because when we talk about marketing of fishes, when we talk about preparation of fish from its raw form to a certain processed form, most countries in the world, but especially when we consider the Asian and Southeast Asian countries, for most of us, a lot of fishing, a lot of aquaculture practices are in an environment where the hygiene is not held up to the best option. So imagine whoever is handling the fish handles without proper gloves, proper eye equipment, proper nasal protection, and that is how they may ingest or they may come in contact with the visceral fluids, etc., of the Streptococcus agalactiae, and if it reaches the alimentary canal, then it can lead up to the infection that I have been talking about. I guess I have answered your question. If more, please ask.
Yeah. And also, Dr. Melba suggests that the audience, if you're interested, you should go to look at the FAO paper on this profile of this bacteria, especially GBS. Okay. So thank you very much. If it's no other questions,
Salwany, can I make a follow-up question?
Okay.
Yes, please.
Yeah, I don't think that, even if you handle the infected fish, if the bacterial load is not that high, and if you don't have any, say for example, wound, I don't think that you can get the infection from fish to human. The only major route that is possible is when you ingest the fish raw that is loaded with this GBS. So I can't see any potential of just handling the fish. You can get the infection GBS.
Okay, while I agree with you that the bacterial load would be very low, that's absolutely true. But when we talk about a hyperinvasive strain like ST283, actually the thing is that infection of GBS through ingestion earlier used to be a very rare occurrence. It used to be so rare that only neonates would be seen positive with this infection, or people who have immunocompromised, or people who are diabetic. Those are the people with comorbidities, etc., were usually reported to have GBS through ingestion. However, after the appearance of ST283, there are confirmed reports that adult humans were absolutely healthy, who did not have any other comorbidity, also got an acute infection to the point where it reached to the stage of endocarditis, meningitis, etc. So because there is a certain hypervirulence to this particular strain and to this particular strain that I have discussed, this angle of fish viscera becoming a potential source of infection was brought up here.
Okay, that is fine for Ed. And I think we thanks for your presentation and hope we can study furthermore on this zoonotic disease. Yeah, thank you.
Thank you so much.